All right, so our next speaker is Dr. Jamie Darwin. Dr. Darwin is currently a postdoctoral fellow in the laboratory of Dr. Stuart Schreiber at the Broad Institute. He's going to talk to us today about compound-mediated interferences in homogeneous proximity assays. Jamie, the floor is yours. Welcome.
Thanks for the intro, Serene, and it's always tough to follow up after a great talk. Thank you, Michelle. So I'll get right to it. Let me just get this out of the way here. So a couple of housekeeping things. So here's my contact information if you have some follow-up questions or want to reach me. And the other thing is I don't have any conflict of interest or financial disclosures. I mention this because I'll be talking about some proprietary technologies. And so I don't want to, I don't get any kind of reimbursement by PerkinElmer for talking about AlphaScreen or whatnot. So I just mentioned that I talk about them just because they're of interest to this community. So today, in the short time we have together, I'm going to divide this talk into three sections. First is some background on homogeneous proximity assays and interferences, and just the underlying technology. And then we can transition that and talk about how compounds themselves might be able to interfere with these assays. And then armed with this knowledge, the fundamental chemistries going on, we can then exploit this to design better assays to prevent enriching for interference compounds, and also to identify any artifacts that may slip through, and also to give you more confidence in pursuing a particular active compound. And so compounds that interfere with our assays, they're significant burdens in drug discovery and chemical biology. So much so, I mean, we're talking about this right now, but the AGM has a very extensive entire section on compound interferences. I encourage you to check out. And some of the ones we heard about in Michelle's talk, you're familiar with. Compounds can interfere with assays by non-specific reactivity. So here's one of my favorite compounds, curcumin. They can react with glutathione. You have aggregators that essentially form these like grease ball-like particles that will non-specifically perturb usually proteins. And then you have compounds that don't really even interact with your biology, but they will absorb or fluoresce light, and they can interfere with light-based readouts, which is, a lot of our HTS technologies involve light readouts. And when you go to higher order assays, like cells, some of these things like reactivity and aggregation can produce tantalizing readouts. For instance, if you're looking for changes in histone acetylation, but it's actually non-specific, and they can lead to toxicities, or in a really high content assay where you don't necessarily know what's causing a particular phenotype, they can produce some really interesting results, but it's actually due to really undesirable mechanisms. And if you bury your head in the sand and don't think about this, you can spend your time and waste resources going after these. And if you really drop the ball, it can lead to flawed science. And then, as we see time and time again, this can be propagated in the literature, in scientific vendors, in patent literature, et cetera. So that's why we're having this talk today. And so to give you a framework of how to proceed, first let's define interference. I have a liberal definition to offer. This is really any compound behavior that will confound your interpretation of your readout, and that's going to prevent you from identifying really the high quality chemical matter you want. And then you can break interferences from compounds down into a couple of categories. There's what technology-related interferences. So this is not interacting with your target. And these are things that will, like, for instance, interact with if you have a affinity tag. It could fluoresce, or it could, bind to your like luciferase system. And importantly, when you do an orthogonal assay, these will typically be inactive. And when you do a experiment for a specific interference, they'll be positive in that screen. This is in contrast to non-technology interferences, where the compounds themselves will actually interact with your biology, your targets, but by a non-specific or poorly optimizable way, like non-specific reactivity or aggregators, cytotoxicity. And importantly, I'll mention this several times, that these can be positive in an orthogonal assay because they're actually changing your biology. And of course, what we're looking for is compounds that generally won't interfere and won't be bioactive from one of these undesirable mechanisms. And of course, things can fall into several categories. And again, you can have compounds that will interfere with technology and still be useful, just as long as you have maybe an orthogonal method, or you've characterized this interference. And things can be cytotoxic or reactive, but it's usually with a specific mechanism in mind. So let's talk about what exactly makes a proximity assay. And so these will involve a donor and an acceptor reagent that, when they're brought into close enough proximity, you generate a signal. And you can make these with a gain or a loss of signal, depending on your configuration. And you can customize these. There's a lot of off-the-shelf reagents. And really, the key thing is that there are no separation steps involved. And that's why we call it homogeneous. You essentially are adding things to the well. There makes it high throughput and amenable to HTS. But the drawback is the compounds are still in the well throughout the assay, and they are available to interfere. And so these proximity assays can be adopted for a variety of biological questions, like PPIs. They can quantify specific analytes, or reaction products, for instance, like post-translational modifications. And so I'll take a little bit of time to describe some of the main players in homogeneous proximity assays. And the first is AlphaScreen. And the way it works is this. If you have your analyte, there'll be two areas that you're going to try to capture. One with a donor bead and some sort of affinity system, and then an acceptor bead at a different location that uses a different capture reagent system. And when these two beads are bound to your analyte, like so, you can excite your well with a high wavelength, low energy laser. And through a series of events within the bead, it'll produce a molecular oxygen in an excited state called singlet oxygen that can diffuse out of the bead. And if it's within sufficient proximity to a nearby acceptor bead, it can continue this cascade of chemical reactions to produce an emission event of photons at a higher energy, lower wavelength light. And so, not to belabor the details of the chemistry, but this is what's going on in each of these beads. And you can imagine that compounds that may contain features like any of these components may interfere, and also that compounds that interact with the singlet oxygen could disrupt your AlphaScreen signal. We'll talk some more about that. But again, it's important just to understand what's going on in each of these beads. Then the other main players in HPAs are the following. There's resonance energy transfer. And most commonly, you'll see this in FRET or TR FRET, where in this PPI assay, for example, you have a donor fluorophore linked to a capture reagent system. And when it's excited at a higher energy, lower wavelength light, the donor fluorophore can be excited and emit photon. And this can essentially be transferred, this energy, to a nearby acceptor fluorophore through this resonance energy transfer phenomena. And if it's in sufficient proximity, it can generate emission at a higher wavelength that you can measure. Relatedly, you can, instead of using a donor fluorophore, you could use a bioluminescent source like a luciferase. That is essentially this luminescence will excite similarly an acceptor fluorophore. You can even harness radio labels that, let's say, a radio labeled substrate for a post-translational modification. The radio label gets incorporated, and if you have a bead with scintillation cocktail within it, like the scintillation proximity assay beads, that this radioactive decay, if it's in sufficient proximity, it can essentially lead to a cascade of events producing light that you can detect. And just for completeness, you see a lot of this more so now with things like nanobit and others, is reporter complementation assays, where you can take something like luciferase and chop it up into two fragments that by themselves aren't active, but if they're brought within sufficient proximity, they generate they complement each other, and the full activity is restored. And you'll see this now, and we use it a lot in clinical chemistry analyzers too, or related technologies. So next, what we can do is, now armed with this knowledge of how these proximity assays work at a general level, and knowing that compound interference can be such a big problem, we can start to think about how compounds would interfere. But again, let me try to give you some more evidence of how big of a problem this can be. So this is some data from my colleague Kamiar Hadi in Germany. And here's an example where he did 4 different alpha screens versus different targets. And among the active compounds, what you'll see here in the red is these are the same compounds that showed up in all 4 of these assays, different targets. And you can see here that in for instance, this assay here, almost all these active compounds were frequent hitters with this technology. So again, if you don't respect the fact that compounds can interfere, you potentially could be, essentially every hit you follow up on could be an artifact. And so that's why you have to take this seriously. And there are a lot of ways compounds can interfere with technology. And so I like to break it into categories. So there'll be 3 categories I'll talk about now. The first one I'll spend the most time on is compounds that attenuate your signal. And this can occur at a couple of different interference points. The first are compounds that interfere with the donor excitation. Others can interfere with the donor to acceptor transmission. And the third is the acceptor emission stage. And they'll all have the common effect of reducing your readout signal. And you'll hear this term a lot with fluorescence quenching, and getting into the various details of how that can happen. I won't belabor it today, but it's in our AGM chapter linked here. So let's use an example of light scattering, where let's say you have a compound that forms a precipitate in your well. And this can happen a lot with screening because generally you're at a higher concentration and these aren't optimized for solubility just yet. And so if you have a light source and it shines through this precipitate, it can scatter the light, and that can attenuate your final readout. So here's a practical example from Philippe over at PerkinElmer, and that's in our AGM chapter, where in this particular compound, you see here that in alpha ELISA and alpha screen shows this concentration response that looks like it would be active. But when you do the same assay, but you add tween to your buffer, the activity is significantly attenuated. And you can rationalize that by the tween being able to break apart the precipitate. And also, when you do another counter screen with these things called omni beads that I'll talk about later, these are beads that have all of the alpha chemistry within one bead. So again, there's activity even with those, albeit less potent. And that suggests that this compound is somehow interfering with the donor or emission light travel. So some tips is, if you suspect a precipitation occurring, take an absorbance spectra and it'll characteristically show this kind of broad exponential decrease that's proportional to wavelength. And you could also add detergents, which are good for several reasons, if your assay tolerates it. And the other thing that you'll talk about now is colored compounds. And while they're pretty to look at, things like bromophenol blue can interfere with alpha in other assays that involve light, because in this example from Perkin-Elmer, whether it's alpha ELISA or alpha screen, whether you have tween or not, and even when you use these beads that have all the chemistry in one bead, you see this very reliable apparent activity. And that again suggests that somehow this compound is interfering with the absorption or the initial or the final light source. And so that's just something to pay attention. And again, with alpha screen in particular, you're going to see this more with these highly colored compounds. And as you recall, just the final emission is 520 to 620 nanometers. So you're looking for your blue and your green, your purple compounds that you'll characteristically see this. And again, something that's not as appreciated as much as I would have thought, alpha screen in particular is susceptible to singlet oxygen interferences. So as you recall, the chemistry of alpha screen, it's going to involve the diffusion of this excited molecular oxygen, singlet oxygen, from the donor to acceptor bead. And anything that would disrupt this transmission would decrease your readout. So there's two main methods that this could happen. You could have a chemical quenching where it'd be a direct reaction of this excited oxygen species with your compound, and you consume the oxygen. The other is a physical quenching where it's an interaction between your compound and this oxygen, and it just returns it to its ground state, and you don't actually consume the oxygen. So here's a real life example of this flavonoid where, again, alpha ELISA or alpha screen, you see this nice apparent activity. Again, when you add the tween, you see attenuation. So it's probably not the most soluble thing. But then when you use these beads with all the alpha chemistry within the one bead, you see no activity. And taken together, that's suggestive that somehow this compound would interfere with this step here, the transmission of single oxygen, because if all the reagents in the omni beads, they're located within one bead, the compound wouldn't be able to interfere with that step. And again, because it's not interfering with the other, the donor or emission lights, you don't see any activity at all. So again, well-designed series of counter screens, you can learn a lot about your potential compounds. So prototypically, single oxygen will oxidize alkenes and heteroatoms. And so probably the med chemists in the audience are like, oh, thanks for nothing, that you basically described like half our collection. So again, there's potentially a lot of interferences with these. And so the next category of interferences that I'm not going to talk extensively about today, but it's in our chapter, are things that attenuate or that augment your signal. And this can occur at several points. One, your initial excitation from your laser can excite the compound itself, and let's say it could fluoresce. The donor to acceptor transfer could excite your compound, and even the final emission event could excite your compound, and they all have the ultimate effect of increasing your readout. And then the third major category with HPA interferences are things that interfere with your capture and affinity reagents. And this could be compounds that disrupt an affinity tag affinity reagent interaction. So examples would be a nickel his tag system, streptavidin biotin, or glutathione GST system. And then another thing that could interfere is if you have an antibody epitope interaction, that potentially could also be disrupted by your compound. So let's give some examples. And so this is some data from Nate Cousins when he was at NCATS. It's in our chapter. So let's say your homogeneous proximity assay utilizes a biotin streptavidin system. You could imagine that a compound that mimics the chemical features of biotin could disrupt this interaction. And so what Nathan did was he used these true hit beads where the donor bead has the streptavidin coated, and then you have an acceptor bead that's biotinylated, and you essentially can mix those two beads together to produce a complete alpha screen signal, independent of your biology. And when Nate added the beads and the compounds together before adding the biotinylated beads, so this nice apparent activity. And yet when he added the compounds after mixing the beads, essentially there's no activity. What that suggests, one, is that these compounds are interfering with the streptavidin biotin system, and it should show you that that's a very strong interaction. So once that bond, that binding between biotin and avidins is made, it's really hard to break it apart. So you can rationalize this interference by looking at some of these common motifs on, let's say, 1 through 4 with biotin here. And so, again, the pretty straightforward counter screen, you can identify these. But I tried looking for, how prevalent is this? And really, in one TR fret, there's an example, it was essentially 0.01%. That may be a little low, I think, but again, this is kind of the idea of potentially how prevalent it could be. The other two main affinity tag systems that you can get interference with are nickel his tag systems and glutathione GST systems. And with the nickel his systems, you can imagine any compound that could chelate the nickel or interact with the imidazole side chains in the histidine tag, like so, could interfere. And there's some references in the slides here. In our chapter, we talk about this more extensively. And more specifically, this data from Kamir Hadian's group in Germany, with this alpha screen here, with these apparent active compounds, when his group did a counter screen with a donor and acceptor beads that involved a GST tag and a his tag, again, they see the same activities. But when they switch to a biotin and his tag system, no activity. And when they use a true hits counter screen, there's no activity. So that, again, that's suggestive that these compounds, it looks like you got some decent activity. It actually is all due to interference with the tags. And something probably that nobody wants to hear, but you should be aware of, are compounds that interfere with your antibodies. And so this is from Jonathan Bale's alpha screen years ago against the MOS histone acetyltransferase. And when he took these active compounds and they did a counter screen where they spiked in their product, so there's no actual enzymatic reaction occurring, he saw that there's this basically 1-to-1 relationship between the primary readout and a counter screen with no activity. Then when they switched to a different antibody and did the same counter screen and the same assay, but with the second antibody, you essentially see all of this artifactual activity go away. looking at the structures of these compounds, it suggested the proposed interference is that this acetamide groups on these compounds were interfering with the antibody epitope interaction because they mimicked the acetylated lysine that was the antibody target. And so, that suggests that it may be worthwhile to do a follow-up assays with a second antibody if you're so lucky to have one. So just to give you an example now, how can we use this knowledge, with our own projects? So let's say we have a PPI assay here, where one of the tags is going to be this Brett system, NanoLuc, and then the other, you're going to have a halo tag system. I'll talk about these in a second. That essentially a halo tag ligand can be specifically attached to this tag. And then you have a luciferin-based substrate that can produce Brett, and then it can excite this halo tag ligand. And then you essentially can quantify the final emission to relate to your PPI. So what interference points could you have? You could have compounds that disrupt this PPI by your really undesired mechanisms like aggregation or nonspecific reactivity or denaturation. You could have compounds that interfere with the NanoLuc enzyme, compounds that interfere with the Brett transmission, even compounds that interfere with the halo tag, the annealing of the ligand to the tag, and this final emission. And why do I mention this halo tag? Well, here's the chemistry going on. You essentially have this primary halogen here that gets specifically displaced and gets added to the halo tag through enzymatic reactions, very specific. But you can imagine a compound may be able to disrupt this potentially. It hasn't been described, but it's just, this is theoretical we're talking about today, just to give you an idea. And then the other thing I talk about is luciferase inhibitors. There's a whole chapter in the AGM about this by Jim Ingles, Doug Auld, and others. So it's just an idea that, you can take an assay and think about it and start to predict how compounds can interfere. Now we go into the last part of our talk, where we can use this knowledge to design better assays and design counter screens to identify these interference compounds. So some basic principles are you can do counter screens for technology interferences, and these will involve what I call target independent configurations. Your idea is you're trying to bypass the biology or the binding event. So you can spike in a product, let's say, or you can switch up some parts of your affinity capture systems. The other thing to do that I won't talk today because there's other talks I've given on this subject on the AGM website and also in specific chapters about, but you also want to do counter screens for non-technology related interferences like aggregation and reactivity. And so interference counter screens, these are designed to specifically characterize the technology interferences. That's in contrast to orthogonal assays where you're trying to confirm the activity by another method, and you're also inherently checking for technology interferences. So some examples of how you could design a counter screen for technology interferences in HPAs. So you could make a special reagent like so, where essentially it's a peptide that's biotinylated and then also has a terminal his tag. And then you could essentially have a donor bead with streptavidin coat and an acceptor bead with a nickel coat. And essentially, you can use this as a way. So if you see when you add compound to this system, and the readout is perturbed, then you potentially have some interference. Another thing would be you could have a biotinylated GST for if you have trying to do a counter screen where you have a GST system. For alpha in particular, you also have some additional options where you can buy beads like true hits. Again, in the Nate Cousins example I mentioned, where the donor bead has a streptavidin coat and you have an acceptor bead that's a biotinylated BSA. And so you don't need a particular intermediary reagent. It just gets right to everything you need to form this alpha screen complex. And then you can also buy these omni beads where all of the alpha screen components are located within the same bead. And so how does this work in practice? And just another reminder. So let's say you have these apparently active compounds and you're worried about these ones that you think may be interfering with the His nickel tag system. You could use the True Hits kit right here. And when you do that, because there's no nickel His tag system, you see that there's no activity in it. So then that suggests that likely the one thing missing between here and here is nickel His tag. And so again, you can start to rule out various interferences with the different components by designing your experiments like so. Another thing you can do, you don't have to necessarily buy anything, is you could do what's called product spiking, where you could purify or purchase the final analyte of interest. So this could be like a phosphorylated peptide if you're working with kinases, for example, or you could let the reaction go first to completion and then without adding your compound. And then you can add your proximity assay reagents, let them equilibrate. And so really nothing should change with the biology. And then you add your compound. And again, you see a change compared to control, you expect some interference. And you could do the same thing with some enzyme-free systems, or for loss of signal assays, you could use omit your enzyme, and an attenuation of your readout would be likely due to some interference because you don't have your enzyme. And so these are talked extensively in other chapters of the AGM, but when we're thinking about non-technology related interferences, you should also do counter screens for those. So like Michelle had mentioned too, DLS. Other things you can do are surrogate assays for AMP-C or MDH. For reactivity, you could use dial-based probes, or you could do HPLC mass spec or fluorescence assays. And again, like Michelle talked about, NMR is a powerful method. I use a lot of this technique called alarm NMR that involves a protein-based thiol probe that specifically looks for non-specific protein reactivity with compounds. Other things you want to think about are metal contamination. So you could spike in a chelating agent like EDTA if your assay allows. You could do redox cycling assays that we've, again, are covered in the AGM. And then others if you're doing cell-based HPAs like cell health and selectivity. And things you can do to mitigate these interferences include detergents, non-ionic detergents when possible, and also decoy proteins like BSA that can help soak up some of the aggregation and also serve as reservoirs for reacting with really hot electrophiles. You could also include scavenging reagents too. Chelating agents if you don't have like a His tag nickel system, and limiting the use of strong reducing agents in excess of, let's say, 1 millimolar. So finally, as you know, we kind of get to the end of the talk, some things I want to talk about. So you want to design and interpret orthogonal assays carefully. And so orthogonal assays, especially with proximity assays, you want to use an independent technology. So avoid shared features like technologies in capture reagent systems. And you should know that orthogonal activity does not mean tractable or useful activity. So you may want to consider a non-homogeneous or a biophysical technique. So let's say you had a primary alpha screen assay. You could do something like a TR-FRET or a BRET as an orthogonal assay. But again, all these are similar enough to your alpha screen. They both involve some sort of fluorescence or light transfer, and may involve similar capture reagents. So um, alternatively, you may want to think about doing, let's say, a mass spec based assay, or let's say you're working with post-translational modifications. You could go old school and do a radiolabeled ligand and do just old-fashioned autoradiography, lower throughput. But if you're really trying to confirm activity, you can do that. You'll see now people will adapt HPAs to cell-based assays. And often this is done where you treat your cells, then you lyse them, and then you are essentially analyzing the lysate. And these are going to be susceptible to cytotoxic effects. So one thing you want to think about is the final readout can be the result of a lot of upstream processes. So let's say you're looking for inhibitors of this acetyltransferase. So you want compounds that reduce this K27AC. And this could be due to inhibition of your target like you want, but it could also be due to a variety of things like changing the turnover of your substrates. It could hit other related enzymes, et cetera, or even just kill cells so there's no histones left. Another thing to think about is if you're doing washing steps, that doesn't necessarily mean you're removing all of your compound. So just because you do a washing step before you start your HPA, you may still have intracellular compound that can interfere. And the other thing is the lytic step can also introduce an interference compound that was extracellular, but upon lysis can mix and now interact with your intracellular components. So you want to be mindful of all the different steps and what they could do. So with HPAs and cell-based homogeneous proximity assays, it's always essential to perform cellular health assays. And you want to think about the proximal and distal components as well, really characterize what's going on. And there's an excellent review by Fabian Vincent and colleagues at Pfizer in Cell Chemical Bio. So you should check that out as well. And the last thought I'll leave you with is you want to think about using known interference compounds to characterize your assay during the development stage. So oftentimes you can't always predict how a particular mechanism of interference will affect an assay readout. So that's what, if you challenge your assay with known interference standards, you can get an idea. So you know what the readout looks like if it's ever encountered. And then the second thing is it allows you to estimate the prevalence of any particular interference. Then you can plan accordingly in your counter screen. So let's say you had an alpha screen and no matter how much of these non-specific electrophiles you added in, essentially the readout wouldn't change. So then you probably have a good idea that your assay isn't going to really enrich for reactive compounds. But then let's say every aggregator you test on it is super active at between 5 micromolars and above. That should, trigger a couple of things. One, you may want to consider adding more detergent or changing the concentration of your enzymes. And the other idea is you may want to then prioritize doing things like DLS on all the active hits. And so just by including, a plate full of these standards, you can get a lot of information, save you a lot of headache later on. So to summary, these interferences from compounds, they're significant, but they're surmountable. I know it may seem otherwise based on the talk, but not every hit is an interference compound, but you need to be critical. There's enough of this stuff going on that we're able to devote a whole talk and a whole section of the AGM to this topic. So that should kind of tell you how important this topic is. Compounds will interfere with the technology itself, but also it will modulate your biology of interest, but by undesired mechanisms. And to reiterate what Michelle said earlier, just like with the biophysical assays, it's always best to do a combination of follow-up assays. And what I recommend is you want to do some technology-related counter screens first to characterize, does your compound interfere with the technology and how much, and then also non-technology related counter screens as well. And then also in combined with orthogonal assays. So avoid the temptation to say, well, I just did an orthogonal assay, I see activity, so we're good to go. Because by characterizing interference by a counter screen, you may be able to determine whether the primary or the orthogonal assay is a better assay to follow up on, for example. And again, just be mindful, orthogonal does not always mean useful. So with that, just acknowledge my mentors here at Broad, Stuart, and Bridget, and then all the colleagues along the way, especially my co-authors on this chapter in the AGM, Jonathan, Doug, Nathan, Kamyar, Philippe, Jared, etc. And then with that, I can look and see if there's any questions. And then I can also, again, feel free to reach out to me by email.
Thank you very much. Thank you very much, Jamie, for a nice talk. There is actually one question for you. This is from Thomas Chung, and he's asking, are omni-beads less sensitive to singlet oxygen quenchers? Since donor acceptors are in close proximity on a mixed bead, the proximity distances are very much lower than the true hit formats.
Yeah, so as far as do I have, great question. So do I have the specific evidence I could show you one way or the other? No, but you're right, because in the omni beads, all the components are within the bead itself. So you essentially, they would be less susceptible because, again, compounds that interfere with single oxygen, they won't be available to disrupt that transmission because they won't get in the polystyrene beads.
I see. Thank you, Jamie. I can't see any more questions. I don't know, Steve, do you see any more questions for Jamie here?
No, I don't see any questions coming in. Can you hear me?
Yes, I can hear you, Steve. Thank you.
Okay. Jamie, I did have one question for, there's a lot of Alpha-Lysa kits available off the shelf, and you commented on OmniBeads versus Alpha-Lysa. I'm just wondering if you can comment on which may be better for the different kits available. And, is there something similar for TR-FRET?
As far as the first question, I wouldn't be able, again, it depends what system you're using. I wouldn't be able to provide, say, I have evidence right now to say which one would be best. It may be empirical. You just have to try. There may be differences in potency with, the IC50, depending on what kit you use, but presumably if it's interfering with the technology, no matter which type of bead you used, you could pick it up. As far as kits for TR-FRET, I don't know of any, but again, a lot of the reagents that you could use, you could design. So if it's involving like a affinity tag system, like a His nickel system, and streptavidin biotin, you could engineer, in your own lab, a peptide, a biotinylated peptide, and kind of bypass the need to buy a kit for, let's say, TR-FRET. I hope that answers your question.
Yes, that's great. Thank you.
Thank you very much, Jamie, again, for this wonderful presentation. There's no further questions for you.